Hello and welcome to our Clinical Genomics webinar for this month. I'm Mariam, the Science Communication and Marketing Officer at GenomeArc, and today I'll be hosting our guest Deniz Duzenli. Deniz is a master's student at Humber College majoring in bioinformatics. We had the pleasure of her joining us as an intern at GenomeArc these past few months, and today she'll be discussing her work on the Pan Cancer Genome from a bioinformatics perspective. This is a topic we got requests for, and we're thrilled to have Deniz discussing it with us today. Thank you so much for joining us today, Deniz.
Thank you. Thank you for having me. I'd like to start off by thanking Mariam, GenomeArc, and Dr. Mohammed Udin Dafil for having me for this talk today. Although I have recently started my path in bioinformatics, I'm very excited to be giving this presentation today, and hopefully I can do the field some justice and provide some interesting insights for everyone here. So let me begin by sharing my screen, and then we can get started. All right, so my talk today is titled Unveiling the Pan Cancer Genome Through a Bioinformatics Approach, and it's just going to be talking about the work that I completed while completing my internship at GenomeArc. So just to give everyone a brief outline, I'm first going to be defining cancer and pan-cancer genes, just so we're all on the same page about it. And then I'm going to move on to the objective of this research and my work at GenomeArc, the data types we used, methods, and then some really interesting results at the end. So everybody may have slightly different definitions of cancer based on their own personal experiences or own personal research that they've done. Excuse me. The National Cancer Institute's definition is that cancer is a disease in which some of the body's cells grow uncontrollably and spread to other parts of the body. Last year, a group of scientists, Brown and colleagues, wanted to kind of challenge this definition and come up with one that explains more of what cancer is at the molecular level. And the definition that they came up with is that cancer is a disease of uncontrolled proliferation by transformed cells subject to evolution by natural selection. So I think both of these definitions, combining both of them, really gives us a nice overview of cancer, what it is and what it does. But ultimately, the cause of cancer is changes in the DNA within our genes, which then lead to mutations. So in 2022 alone, there were 20 million new diagnoses and 9.7 million deaths. So I think we can all kind of agree that we do have still a lot of work to do in the cancer research field to better understand this disease and to help lower these statistics in the future. There are two different mutation types that I'm going to be explaining just to help with the understanding for any of those that may not be familiar. So there are germline mutations, and these mutations are passed on to offspring through their parents. So either one of the parental gametes or both of them may carry these mutations, and that gets directly passed on to the embryo, and then that individual can also continue to pass it off to their offspring. Somatic mutations, however, are acquired, so these can happen anytime after birth during the person's lifespan. These can be caused due to environmental stresses, they can be caused by illnesses, they can be caused by any damage to the molecular mechanisms, DNA replication or repair, and these tend to just stay within the individual and not necessarily passed on to offspring. Pan-cancer genes, which are going to be the focus of today's presentation, are commonly mutated in many different cancers. So these genes, as you can see in the image here, one gene can cause many different cancers, regardless of whether it be germline or through germline or somatic mutations. And these are very, very important to study because it can give us a comprehensive view and very good insight into how cancer develops, how it progresses, and just helps us get a better understanding in less amount of research time about cancer and these different genes. So at GenomeArc, the main focus of the work, and mine as well during my internship, was somatic pan-cancer genes. So these can help identify common mutations. As I mentioned in the previous slide, pan-cancer genes are very important to study just due to the fact that they are found in many different genes and cancers. So this can help identify common mutations, pathways, variants, to help us better understand cancer and how it functions. Starting to sound like a broken record, but the ability to study many cancers at once, very important. I'm going to be reiterating this throughout the presentation. This helps more affected people because we can better understand the disease that they're going through, how to better treat them, and how to prevent that cancer from coming back. And there's a higher possibility of identifying common patterns or pathways, which can then in turn reduce research time and the use and development of multiple drugs. So although this may not be the best outcome for pharmaceutical companies, ultimately it is the best outcome for the patient, because we want to simplify their treatment plan as well as prevent any negative side effects or any drug-drug interactions from having a very complicated treatment plan. So the main objective of all of this work is to identify pan-cancer genes by analyzing large-scale tumor genomic data. This work is also going to be implemented into a platform at GenomeArc called Horizon, which is being developed to have a comprehensive overview of these variants and genomic variant analysis. And it is a groundbreaking tool that I hope to explain further to get all of you excited about it as much as I am excited for the development of it. So moving into the data types that we used, we decided to go with exome and whole genome data. This is just to provide a very comprehensive overview of the genomic data that's being used and help get very detailed results. And we wanted to use tumor and normal matched data as well, because this helps us to directly compare between the healthy and the tumor data and samples, as well as help reduce the number of false positives that may exist. All of this data is taken from 2019 to present year, and they were all extracted from the publicly available database CBio portal. From all of these results, from all of the data sets that we used, we found a total of 27 different cancer types. At the top of the list, we have colon cancer. So this list is in decreasing order of how many different genes these cancers were found in. So at the top of the list, we have colon cancer. It was found in 41 different genes. And then at the bottom here we have myoepithelial carcinomas of soft tissue. So this was found in just, variants were found in just one gene type. So we have a good variety here. Colon cancer, we have prostate and breast cancer, bladder, pediatric brain cancer, AML. So we are hoping that this provides a very broad and nice overview of all the different cancer type, or of all the data and results that we have that I will be sharing in the later slides. So for methods, we used GenomeArc's Horizon for variant annotation from HG37 to HG38. So HG37 was the form that this data was in on the CBio portal data sets that we collected. And then HG38 is just the updated representation of the human genome. So we really wanted to have the most up-to-date and recent information and results provided. For the bioinformatics aspect, I mainly used RStudio packages for data wrangling and visualization. These include dplyr, tidyverse, and ggplot2. And then the cancer names that we originally had collected were slightly adjusted just for a little bit more cohesion. So for example, we had metaplastic breast cancer, which is just a more rare aggressive form of breast cancer. And we just grouped it under breast cancer altogether. This just provides a little more cohesion. It's not going to play with the results very much or change anything. Same with the some of the mutations. We group them into loss of function. So this includes frame shift deletion, substitution, stop gain, splicing. All of these were kind of put together into loss of function to allow for more cohesion and kind of get rid of that background noise. And then moving on, where we collected the main results were from the burden values that we calculated and the creation of the statistics tables from this. So these I will be explaining in the next slide. So for the burden calculations, generally this is just looking at kind of the proportion that all of these variants and the different genes are coming up in our results. So we decided to use 3 different types of these burden calculations. The first one is exonic burden. For this one, all of the factors that we have are being used. So we have the variants, we have genes, and we have the cancer types as well. So for this one, it provides the most detailed results and the most amount of information. Moving on, we have non-synonymous burden, and this is just removing all of the synonymous variants. So these variants are kind of like silent mutations. It doesn't really change much at the amino acid and protein level. So we want to have results that remove these and look more at the variants that are causing the different mutations and causing the different issues at the protein and amino acid level. And for these results, we did not include variant types. So it only looks at genes, and then kind of the variant types are all grouped all together. And then finally, excuse me, finally, we have background burden. So this removes the variant types, just like the non-synonymous burden, and only calculates how many variants in total each gene has in each cancer. So this is kind of the more broad one, not as detailed as the other ones, but still provides some very useful information. So whichever equation you would want to use really depends on the research that you're doing and the questions that you would like answered. Since our work is going to be integrated into the Horizon platform that GenomeArc is working on, we really wanted to include as much as we could because it is going to be a very comprehensive tool, and we want to have as many different results as we can so that it can be applied to as many different applications as possible. Moving on to results, let me go back to the previous slide. For each of these 3 different burden calculations, I'm going to be showing results for the top 20 highest burden value results or entries, and then also the bottom 20. So I'm going to be showing you some from the highest at the top of the list and then at the bottom of the list. So we're going to be going through 6 different plots in the next 6 slides and go through all of these results together. Starting off with exonic burden counts, we have the genes on the x-axis, the burden value on the y-axis, and then all of the different colored points are colored by cancer type. You can see on the right side of the screen here. So if we take a look, we have a good number of genes from the top 20 entries of this list. And then we can see here, there's EGFR, CRAS, MUC16, TP53, and TTN. We can see that TP53 has the highest burden value, and then following close behind it is EGFR, and these are both found in lung adenocarcinoma. Taking a general look at the patterns here, we can see that there are a lot of data points for lung adenocarcinoma. And also, I would like to point out TTN, which is just the most furthest right gene on this axis here. And this is found in the most, or there are variants in the most number of cancers here. So we can see that there's 4 different cancer types for TTN, which is, and then following closely behind is MUC16 with 3 different cancer types. Going into the bottom 20 entries in this list that we were able to get results for, all of them had the same burden value. So I believe it was 1.27 to the power of -6. So very, very small numbers. And from these last 20 entries, actually, there were only 3 genes, A1BG, A1CF, and A2M that were here. But we can see that the cancer type list is much longer than the previous graph, which does suggest that although these burden values may be the same and there's not very many genes from the bottom portion of the list, they are found in a lot of different cancer types, especially A1CF here. We can see it has a lot of different cancer types. So this is just highlighting that although these genes are more at the bottom of the list, the proportion may not be as great as the ones at the top of the list. They still might be very good candidates to study because they are found in so many different types of cancers. Moving on to non-synonymous burden counts. So this was the calculation that we had by removing those silent mutations. This one, we can see the similar genes are coming up as the first plot. We've got EGFR, KRAS, MUC16, TP53, TTN. Once again, if I draw your attention to TTN, it's there are variants in 5 different cancer types. And this is just to reiterate, this is only from the top 20 entries of this results table with thousands and thousands of results. So we can kind of take this pattern. This is kind of like a screenshot or a snapshot of the greater patterns that are being found in these results. Once again, TP53 is right up there with the highest burden value, following really closely afterwards EGFR. And we can once again see that lung adenocarcinoma seems to be the most, or seems to have the most number of variants in different genes in this plot as well. For the bottom 20 entries, the lowest burden counts for non-synonymous, for the non-synonymous calculation. We have a few more genes in this list. Once again, we have A1CF there with a lot of cancer types. A2M and A2ML1, we see that they are both just found, or they have variants in 1 cancer type each. So this may suggest that maybe these are not the most focused genes that people want to be studying or looking at, just because especially for pan-cancer studies, because they're not found in very many. But this can, obviously, it can be more useful to certain people depending on the research questions you're looking to answer. But we can see here that, as I mentioned, A1CF seems to be coming up in a lot of different cancer types, A4GAL2, A4GNT. So this is, although these are, again, at the same level of the burden value, they are being found in a lot of different cancer types. And then moving on to the final calculation, we have the background burden. So this is where we are just looking at the genes and the cancer types. So for this one, similar results: EGFR, KRAS, MUC16, TP53, TTN. There are other, maybe like not as well-known genes scattered in there. And then similar results to the other ones. TTN has a lot of different cancer types that has variants in. TP53 and EGFR at the top of the list. MUC16 also has quite a few, or cancer types that it has variants in. And then moving on to the final slide for lowest entries for burden counts, we can see here again A2ML1, A2M. It has the lowest. It's found in the least amount of cancer types here, but obviously these are still very useful. They can still be very good candidate genes to study based on the research questions that you may have. We see here A1CF. Once again, it's found in 4 different cancer types. So these results just really provide a good overview of the general patterns and the general data that we can extract from the work that has been done. These can help narrow down the research questions that people may have and help give them a tool to easily be able to answer the questions that they may have for their research, or even healthcare professionals when diagnosing their patients. From all of these results, I was able to create a list of the top 50 pan-cancer genes, so the ones that are found in the most number of cancer types. Here, I've included the top 3 and bottom 3 from the list, just to provide a little insight for both sides. MUC16 and TTN were at the top of the list. They were both found in 18 cancer types each, and this is not very surprising looking at the results from the plots that I just presented. These were coming up a lot in a lot of different cancer types, especially TTN. TP53, which is a very well-known gene and well-studied, it was in a close second at 17 cancer types, which is, once again, not very surprising, as we do know a lot about this gene. We know it has a big role in cancer. And then at the bottom of the list, we have RB1 and LRP2. These were both found in 5 cancer types each, and then FLG, which was found in 6 cancer types. And this isn't to say that certain genes may be more important or better to study than others. This just kind of provides a nice overview. All of these genes can be very good candidates to study in pan-cancer studies and really help answer all of the questions that we may be having and cut those research times down quite a bit by being able to study many different cancer types at a single time in one project. I'm now going to be focusing on 2 of the genes from the results today. So TP53, a very well-known gene, very well studied, as I just mentioned. This is a pan-cancer tumor suppressor protein. So this gene encodes a tumor suppressor protein containing transcriptional activation, DNA binding, and oligomerization domains. And the encoded protein responds to diverse cellular stresses to regulate expression of target genes and thereby inducing cell cycle arrest, apoptosis, DNA repair, things like that. So if we think about what would happen if there were mutations in this gene, obviously these pathways for apoptosis or DNA repair would not be able to occur. And this is what can lead to the development and progression of cancer, because those cancer cells are not being killed off and they're being allowed to proliferate in the body. This is just a little graph to highlight the importance of TP53. This is just looking at loss of function variants. We have variant count on the x-axis here, and then the cancer types on the y-axis. We can see, this is not surprising because of the results we were seeing in the graphs, lung adenocarcinoma has over 400 different variants. But if we take a look at the other ones too, there are a lot that are closer to 100 or even 50 that have a lot of variants in a bunch of different cancers. So very important gene to study and to understand. However, TTN was found to maybe not be as well understood or studied of a gene, but we have come to the conclusion with our results that it is a very good candidate pan-cancer gene. So the protein product of this gene is divided into 2 regions, an N-terminal I band and C-terminal A band. And the TTN truncating mutations were firstly proven strongly correlated to muscular diseases, which but not in cancer, which is why people may not know about the role it plays in cancer or its importance and not be studying it as much. But looking at our results that I was highlighting earlier on, there is substantial evidence that TTN is a pan-cancer gene. just because of how many times we were seeing it come up and how many different variants it was found in, as well as being at the top of the list for being found in 18 different cancer types. This is just a similar plot to the TP53 one that I just showed. This is, once again, the loss of function variants. We have the variant counts on the x-axis, and although there is not like the drastic over 400 variants as there was in TP53, we can still see that there are quite a few different variants or quite a few different cancer types with a lot of variants. So combining this with the results that we were seeing in the plots, as well as being at the top of the pan cancer list, TTN is a very good candidate gene that we believe should be studied more to understand its role in cancer. So the main takeaways of today's presentation: obviously, cancer affects individuals worldwide, and we need solutions to help combat this. And one of these solutions could be through bioinformatics. It can help us improve diagnoses, treatments, understanding of cancer and its pathways. Bioinformatics is a really interesting field. Like I mentioned at the beginning of my presentation, I've newly entered into. But it's able to take messy data and convert it into really nice tools and comprehensive tools that we can extract useful information from. So bioinformatics is a very good path into understanding cancer and helping work towards a cure. And then once again, I wanted to highlight GenomeArc's Horizon platform, which is a cutting-edge solution providing rapid and comprehensive precise variant interpretation. So I'm very excited for this platform to be released. During its development, it's been very exciting to see the stages that it has been able to reach. So I'd like to thank everybody for listening to my presentation today. If you have any questions at all, I have included the QR code to my LinkedIn here. You can scan that. Feel free to message me or connect with me on LinkedIn. And I've also included GenomeArc's page as well. I highly, highly recommend following their LinkedIn page. There's always new updates in genomic medicine, precision medicine, even with the Horizon developments. And I've also included the GenomeArc website here for any of those that may be interested in any blog posts or any updates, or to dig around the website a little bit. But that brings me to the end of my presentation, and I would like to thank everybody for joining me today. Thank you.
Thank you so much, Deniz. Your talk was amazing. It's amazing to see how much insights can come from data, and also how much, cancer is very diverse, but you've really found like a common lead. And so I have a couple of questions. So first of all, from your analysis, do you think, especially with the potential from the Titan gene, do you think a set of genes can be screened for many types of cancer?
I think definitely yes, especially we can see with the results where there are a lot of pan-cancer genes that do exist, and pan-cancer studies can be very useful in studying different genes for different cancer types. It would, looking at the results like the MUC16, excuse me, the MUC16, TTN, TP53, these might be good genes to study, but as I mentioned in my presentation as well, that's not to say that the other set of genes is not important. It would really have to be like a collaborative, collective work to get those results. But definitely, I think pan-cancer studies should be the future of cancer, or at least a part of the future of cancer research.
Yeah, and do you think do you think in the future of precision medicine, should a drug be designed targeting the variants or targeting the genes, especially since you showed like the amount of variants in the gene, and the amount and the genes itself, how much they're implicated in different cancers?
I definitely think variants. Targeting genes may not be as specific. We want to definitely target variants because we want to, there are, like you said, there's lots of different variants that were found in the results. So to get as precise as possible, as specific as possible, definitely I think targeting those variants is what we want to work towards and what the goal should be.
Yes, I agree as well, honestly. I think variants are more specific and you're really going to the root of the problem. So do you think precision medicine approaches will cure cancer in the near future? That's a big expectation, but like, do you think it could happen?
That's kind of what we're all hoping for, right? We want to be able to have this research lead to cures. That's, I think, what everyone's really working towards. I think it definitely has potential because precision medicine, like it's really going to I'm going to use the same words you used just a moment ago, like to the root of the problem. It looks at the very specifics. So I think getting as specific and precise as possible, I think that definitely does have a big role in potentially leading to cures. Not sure how long that will be, but I think with the technology and the research that the tools we have today, as well as like what is still being developed, there is definitely potential there for cures.
Yeah, that sounds hopeful, and I agree totally. I think research in this area is amazing. Your talk was amazing today.
Yeah.
I want to say thank you so much for joining us and for joining us as an intern at GenomeArc. It was a pleasure having you with us today in these past few months, and thanks so much.
Thank you. Thank you to you too, and to GenomeArc.